A very good morning to one and all present. Now, today's topic is structural alterations in chromosome. Now, before understanding the structural alterations, let us consider that what a chromosome is basically. So, we say that a chromosome is the most condensed form of the genetic material in the form of DNA that lies at the metaphase plate, and it is there during the cell division. Now, this chromosome has importance because all the genetic material that is there in the nucleus can be seen easily under the microscope using these structural units that we call as the chromosomes. Now, depending upon the type of chromosome, we can say that we can categorize the chromosomes into metacentric chromosomes, where the two chromatids they are adhered with the centromere and they are of equal length. While on the other hand, if the centromere is lying on either of the two ends, then we call it as the submetacentric chromosomes. Similarly, telocentric if the centromere is at the telomeric end, that is at the ends, and acrocentric if the centromere is lacking. Now let us consider the example of metacentric chromosome. Now here the structural alterations that are possible, the first important among them is the deletion. Now in the process of deletion, what happens is that a part of the chromosome is basically deleted. So we can see that this part of the chromosome, it has been deleted in this chromosome. So this kind of aberrations or alterations in the chromosomes are generally called as deletions. Now similarly, we have another type of structural alteration in chromosome that we call as the addition. Now in the addition, what happens is that a chromosomal part from some other chromosome may get attached to either of the ends, and we have additional chromosomal part getting adhered to our chromosome of interest. So we say that it's a kind of an addition. That's why this structural alteration is called as the addition. Now, the third important type of structural alteration is called as the inversion. Now, in inversion, what happens is that a part of the chromosomal segment, it gets just flipped and then it gets incorporated. So we can say that it's just the cut, reverse, and then pasting up of the things. So here the inversion, we can categorize it into two basic types. The first that we have just discussed is without the involvement of the centromere, and that is called as the paracentric inversion. That is, the centromere is not involved at all. But on the other hand, we have another kind of inversion in which the centromeric part is also involved, and that is called as the pericentric inversion. Now, the fourth important type of structural alteration in chromosome is called as the translocation. Now, as the word suggests, translocation means the movement from one end to the another end. So we say it is either within the chromosome, there is a movement of one part of the chromosome to the another, or it may be from some other chromosome, the movement to the chromosome of interest. Thank you.